So this expedition represents a unique opportunity to investigate a deep sea environment that's been on the bottom for over 110, almost 110 years. And so we have a number of questions that we're going to ask in this expedition. There have been a few previous reports on the biology on and around this site, but we plan a more comprehensive study of the site's biodiversity. We'll continue also our complementary archaeological investigations. And as we investigate, the wreck, we'll ask these questions about, how does the biology of the wreck differ from the surrounding muddy plains? And most of the area on the deep sea are flat, muddy sort of areas. How do these wrecks differ from that? What animals do they attract that require this kind of substrate? Are any of the changes that we see to the bottom positive or negative, or perhaps neutral? We're going to hear more about our environmental DNA work in a few minutes, but we're also interested in how this new tool compares to our more traditional ways of sampling the bottom. We're going to measure oceanographic conditions, and those, of course, can have an impact on the biology, particularly currents and bottom temperature. And we have some data on other deep shipwrecks, and of course, the data we collect will go into an archive and will allow us to start comparing different kinds of shipwreck environments and those impacts. So to address these questions, we have a number of objectives, and we have to keep in mind that each dive the submersible makes will have multiple tasks to accomplish. So our goal is to describe, to identify and count the fauna on and near the wreck. This is sometimes quite difficult in the deep sea. We want to document how these animals use the various habitats. And we're going to construct what's called a geographic information system, which allows us to map a variety of features of the bottom and the biology. We can overlay different layers of electronic data on maps, physical and biological data. It's a very powerful tool to visually assess scientific data. We're also, as I mentioned, we're going to enhance our visual observations with the DNA analysis. We'll be collecting oceanographic data, as I said, and we'll be testing some new oceanographic instruments. We're going to collect data on water chemistry, which will allow us to add to a larger database to try to evaluate how the ocean is changing. And this is in particular in relation to ocean acidification. And I think Anna's going to mention our modeling work, which uses oceanographic data as well as our information on the biology of the wreck. So we'll try to model the dispersal of various organisms. So from past expeditions and our own knowledge, we have some idea of the animals that we expect to find on the wreck. But always with deep sea work, we'll be making new discoveries. Some of the animals we observe are only known from dead specimens that perhaps occur just in museums. We'll be viewing animals in their life situation, maybe for the first time. We'll be able to describe their colors and behaviors. It's also very possible that we'll be observing some species that are new to science. That's not uncommon in deep sea work. We'll be recording the distributions of the animals, their use of habitat, their behaviors, and their modes of feeding. So finally, what facilitates this work are our expedition tools, makes this work possible. We have, of course, the 5-person deep submersible Titan, various oceanographic instruments, and our eDNA tool. Our sampling is going to be, or our views of the wreck is going to be non-destructive and of low impact. Yet the submersible and its high definition cameras and instruments will allow us to collect a significant amount of scientific data. And one of the things that's common in these sorts of expeditions is our team will be working on these data for long after this expedition is completed. So it'll have quite an archive to keep us busy for a while. So with that, I'll turn the briefing back over to Stockton.
Well, thank you very much, Steve. And with that, I'd like to pass the baton over to Dr. Anna Gebrick, who will talk a little bit more about the biology.
Excellent. Thanks so much, Stockton. And thank you, Steve, for the great overview of the scientific plans. And now I'm going to talk a little bit more about how we're planning to link the data that we gained from Titanic to broader research that is happening in the Northern Atlantic and beyond that as well. Can we get yes, thank you. So studying the biological communities of Titanic wreck will allow us not only to gain new and important data on local biodiversity of the site, but it will also contribute towards a more holistic understanding of the biological and also oceanographic processes in the entire Atlantic Ocean. So one example is that the deep sea corals that are seen on the wreck that you could have spotted on the slide that Steve was showing earlier, those are living organisms with complex life cycles. And when they spawn, they release small larvae that shoot into the water column just like small balloons and travel long distances with the ocean currents. On the screen now, you can see the first outputs of a biophysical model that predicts how a particle that is released from Titanic site could spread within just 30 days. Collecting biological and oceanographic data from the Titanic site would allow us to significantly strengthen those models because those are just theoretic, but getting actual measurements would be really helpful to improve the quality of these models and understand better the dispersal of cold water larvae both from and to the wreck. And ultimately, that would help us to answer a very important ecological question: to what extent could the Titanic wreck act as a source of larvae for new populations of cold water corals in the Atlantic? Can we get the next slide? And in general, linking the data and knowledge gained from Titanic sites with broader research in the Atlantic Ocean and beyond, contributing to the diverse knowledge system, is one of the core values of expedition, really. And one example of that is the new partnership with the iAtlantic, a large multidisciplinary research program that seeks to assess the health of deep sea and open ocean ecosystems across the full span of the Atlantic Ocean, including around the Titanic site. The iAtlantic Consortium is a large partnership with over 38 research organizations from Europe, Brazil, South Africa, Argentina, Canada, and the USA, and even more associated partners, some of which you can see on the slide. Can we get the next slide? And the final thing that I wanted to highlight, that it is also our commitment to ensure that the scientific data from expeditions is openly accessible to any researchers around the world who are interested in Titanic, or not even researchers, general public, anyone who wants to get access. And to ensure that, we will be using the best practices in long-term environmental data management with the support of the iAtlantic partners to systematically annotate, structure, and upload data to open source repositories. In case of the environmental data, the one called Pangea. Um, I think I will stop now and hand back over to Stockton. Thank you.
Thank you very much, Anna. Next person to present will be Beverly McClanahan at eDNAtec, an amazing St. John's based company, and she'll tell us a little bit more about what's going to happen with these water samples that we will be taking. Beverly.
Thank you for the introduction, Stockton, and hello, everyone. As we heard from Steve, we'll be using environmental DNA analysis to study the biodiversity in the deep sea at the Titanic wreck. And I'll give you an overview now of what environmental DNA is. Environmental DNA, also known as eDNA for short, is DNA that has been shed by organisms into their environment. So just by swimming, breathing, and moving around in the environment, every organism leaves a little bit of DNA behind. From whales and fish to little sea snails and plankton, they all leave fragments of DNA behind. And these fragments contain unique signatures that can be used to identify the species that the DNA came from. So we can collect a non-invasive sample of water that contains DNA from all the organisms living in that environment and use DNA sequencing to reveal the total biodiversity in the sample. And eDNA is really transforming how we characterize ecosystems because we can detect total biodiversity from environmental samples where previously we would have to employ multiple methods to survey a whole ecosystem. And the biodiversity data that's generated using eDNA on this expedition will be used to support those scientific objectives that Steve outlined earlier. Moving on to the next slide, I'm just going to talk a little bit about where I'm joining the expedition from. I'm an ecologist with eDNAtec, and we are a company that specializes in using environmental DNA. So we're using DNA sequencing to transform how we characterize and monitor ecosystems to support environmental stewardship of both marine and terrestrial environments. And we have a dedicated ISO certified laboratory facility that provides end-to-end support in using environmental DNA. So right from field sample collection through the entire lab process to data analysis. And this facility is located in St. John's, Newfoundland, and this is where the samples collected on this expedition will be processed. The lab is equipped with the most advanced DNA sequencing platforms, and we have a scientific and technical team with expertise in all stages of the workflow. So ecology, genomics, laboratory science, and bioinformatics. And eDNAtec has conducted hundreds of projects for clients around the world, and we work really closely with various ocean industries, including energy and fisheries, to bring eDNA technologies to environmental assessment and monitoring. This Titanic expedition will be one of the deepest and most ambitious studies that we have undertaken, and we're really excited to be working with the team here today and to use this opportunity to learn more about the deep sea. And I'll hand it over to you, Stockton.
Very good. Thank you, Beverly. Well, that's great. Now, a couple of questions have come in. The first question I see here is, why is it important to study the deep sea? How is it different from other areas of the ocean? Well, that's for you guys. Steve, you want to take the first shot?
Yes, I had to unmute my microphone. Yeah, I'll make a couple of comments about that, and perhaps Anna or Beverly can join in. This is the deep sea is probably the largest ecosystem on Earth, and yet it's one of the ones that's least well known. So it has a great impact on the world's climate and the world's biology. The changing ocean, changing acidification, changing temperature patterns have an impact on oceanographic currents, which are deep, which have an impact on upwelling of nutrients, which then impacts plankton blooms. So there's a lot of connectivity that people don't think about when they think about one part of the ocean. So, we're part of a worldwide effort to gain more data on about the deep sea. Anna and Beverly, feel free to add to that.
Thank you, Steve. I will just echo what you've said and also throw in a scientific term that is called bentopelagic coupling. And what this term really refers to is how the ecosystems of the seafloor are connected to the water surface, to pelagic ecosystems. We observe a lot of the change, including effects of the climate change on the surface waters, retreat of glaciers, sea ice, things like that. And we might not always be able to see things that are happening on the seafloor, but we know that the two components are very strongly connected, and things that are received by the seafloor from the surface that are stored in the deep sea or released from the deep sea, they are all connected to each other through the trophic levels and through this bentopelagic coupling. So yeah, definitely studying the deep sea ecosystems is incredibly important to understand this whole ecosystem processes, especially now when a lot of the deep sea is not covered by any national jurisdictions. So those are areas beyond national jurisdictions, and understanding them is really important to protect them.
Great. Well, our second question is, why are shipwrecks important habitat and important to study?
Okay. So most of the deep sea is relatively sandy, muddy, flat plains, essentially. That doesn't mean they're devoid of life. There's a lot going on the sandy, soft substrate bottom. But shipwrecks and other hard structures represent islands of biodiversity. They create a different kind of habitat in a place where that habitat maybe didn't exist. And they attract animals. They attract anemones and corals that settle on the wreck and stay there their whole lives. They attract a variety of fish and more mobile animals, octopus, squids that use the wreck opportunistically. So it represents potentially an enhancement to the seafloor in terms of habitat and productivity.
Great. I'd throw another wrinkle on this thing. One of the things that makes shipwrecks great, I mean, obviously, as Steve was saying, you generally know when they went down within reason. So different than a boulder or something. But from an OceanGate expedition business perspective, one of the challenges to deep ocean exploration is the cost, and it's very difficult. Huge need and a huge cost, and governments and philanthropic entities can only do so much. And one of the great things about shipwrecks is they make for great storytelling. And one of the ways that we're able to support this kind of scientific research is by finding different ways to fund it. And so from OceanGate's expeditions perspective, we can take media, as we'll do this year and we did last year, and film these wrecks and these locations. And we can bring people who are willing to help fund the operation to participate. And that gives us a completely different way to fund this and be able to go back to the Titanic and other sites every year, which is very unusual in the deep ocean. So another question is the deep sea affected by changing Earth climate?
Well, yeah, we mentioned that. I think Anna will, and Beverly can chime in again. Most definitely, it's being influenced, and because it's such a massive habitat, what would appear to us to be small changes in temperature or salinity or acidity have a massive impact, because we're talking about such a huge volume of water. So, definitely this is one of the things we're concerned about is our changes in the deep sea, and they're certainly happening.
Right.
Absolutely. Just one more opportunity to highlight here how important it is, really the exciting opportunity that we have to start building the time series, because we do know that the deep sea systems are impacted, but the extent of direct impacts is really hard to measure when we don't have continuous observations. And expeditions like this give a fantastic opportunity to do repeated measurements over the years, which will hopefully help us to understand those impacts better.
Great, and that sort of ties into this next question, which is question was, what makes the study on and around the Titanic unique or different from studying other shipwrecks? And I look at it from a business and an operational perspective, which is, it's very difficult to get funding to go back to the same site. There's so much to be explored in the deep ocean that if you look at most research organizations, other than a few hydrothermal vent sites, there's always something new to go look at. And the idea that you would go to the exact same site every year at roughly the same time with similar equipment is very unique, and only the Titanic draws that kind of following to allow us to be able to afford to do this. We hope to do other sites like the Bismarck and things like that in the future, but the Titanic is the thing that everyone's very excited to see.
Yeah, that's a very good point, and I can echo that. That's from a scientific perspective and archaeological, almost any view, going back to the same spot, multiple times has huge value, and we rarely get to do that in the deep sea.
I just want to add one more perspective here, which is something that is called restoration ecology, which is linked to the slides when we were talking about the models of larvae dispersal. Because when we look at biodiversity of Titanic, we can look at it from different angles. And one of the novel approaches is to think about, okay, how does the biodiversity of Titanic feeds back to the ecosystems surrounding it? And this release of larvae of deep sea corals, which are very rare and important habitat building organisms, is a very important impact. And those types of studies, again, can only be conducted if we continuously monitor the oceanographic conditions and the organisms that live on the wreck.
Very good. The question is, do you expect any surprises on the mission? Do you expect any surprise discoveries?
I think the simple answer is yes. One of the things I always say is I've never every dive and expedition I've made to the deep sea has had something new, whether it was small or large. There's always something new, because a region that's just so poorly explored. We'll see fish we've never seen before, perhaps colors and behaviors we've never seen before. So that's one of the things that makes it so exciting.
Great. A question, are there complications to conducting this work around a historic wreck site? Can you address concerns that may exist about how you get this work done without disturbing the wreck? Yes, this is a World Heritage Site, the Titanic. It's a grave site where over 1,700 people died. We do not pick anything up. In fact, the sub does not have a manipulator currently, so we can't pick up artifacts. We have no intention of doing that. There have been over 5,500 artifacts taken from the site over the years, but that's not the purpose of what we do. We're going to go down and we're going to photograph, we'll take a water sample is all, which shouldn't disturb anything. And we're very careful not to touch the wreck. We don't go inside it. And we in fact have a small memorial service every mission that we go out there to make sure we remember that there were many people died at this site. And our hope is to validate what happened by getting as much knowledge as we can and keeping interest in the story of the Titanic. So next question is, can you recap the research that was done during last year's expedition and whether there have been any publications yet, or was last year's expedition a shakedown cruise research-wise? I'll hit one piece of that. Unfortunately, Dr. Bridget Buxton was out. Steve had medical problems and wasn't able to come out last year. There will be research papers coming out on that. That takes a while to assemble. And it was also a bit of a shakedown cruise. We did have a lot of technical challenges that we think we won't have this year. We had weather challenges, we had COVID challenges, so there's a lot of that stuff. But we still got the best imagery ever taken. And Steve, maybe you can talk about what you were able to see looking at some of the 4K images that we brought back.
We haven't put out publications yet, but what we intend is for last year's data to become, part of the data we analyze from this year. So essentially we'll have 2 years worth of video data to analyze and observations, from the wreck. So the first year, while it was a shakedown in many ways, still produced significant data that we can use with the upcoming information. So it makes it, much more powerful data package. The images were great. We saw one of the things that came to mind in looking through the past data is, and related to the coral modeling, is we saw these small corals in a variety of locations around the shipwreck. And that was some new information, and we hope to follow up on that to look at the distribution of corals on the wreck.
And one of the things that our goals with the Titanic expeditions that we're doing is to answer a very common question, which is how long will the wreck be there? And my standard answer is going to be there for a long time. How long will it look like a shipwreck is really the question I think most people are asking, because it will slowly decay. And every time we go, we're taking these images. So when we go back this year, we may be able to see areas where maybe a section of the promenade deck has fallen forward, as it's tended to do. Some of the samples, the site has only been visited, it was once in 2019 very briefly, and then 2010. So it's been a long time, and it's been over 2 decades, and I think only once was it visited 2 years in a row. So it's going to be interesting to see just on a year-to-year basis, when things, do they gradually go or do they collapse all at once? We have a question, how will the eDNA samples be collected and stored? I think this one's for Beverly.
Yeah. So we'll be collecting water samples for eDNA analysis using Niskin bottles, which are these tube-shaped devices that can be closed at a particular depth and location to collect a water sample. And we'll have a couple of those bottles attached to the exterior of the Titan submersible, which can then be closed at our target sampling locations around the wreck. And then back on the vessel at the surface, the water samples will be filtered to capture DNA from the water. And then the filters, which now contain the DNA, are frozen to preserve the DNA and prevent that DNA from breaking down until we can get it into our lab to continue processing.
Great, thank you. I have a question here. How important is your research in terms of understanding that and what to expect in the near and long term? I'll take the first step, but then I'll let Steve and Anna follow up. One of the things I think is most important about what we're doing is this idea that it's a different way of doing deep sea marine research. So, it allows, by making it all be privately funded, we can be more nimble. We can go to places. There wouldn't be a lot of support, I think, for there certainly isn't support for an annual visit to the Titanic, given all the other scientific needs for the very few deep sea research vessels that are out there. So this is going to make this a very unique opportunity, and we hope allow us to expand and do many other sites where we can bring in multidisciplinary researchers in a format that doesn't take years of grant requests and other shuffling to take place. But I'll let Steve and Anna take it from there.
Now, one of the things that comes to mind to me is that between all of us, Murray Roberts' lab, my work over 2 or 3 decades, and Anna's work, all of this builds and supports each other. We're collecting data with a lot of consistency over the years. The way we analyze video data, the way we collect water samples, this allows us to compare, now perhaps a 20-year database on deep sea research that just our group has done. And it allows us to tap into the work that other people are doing as well. So while it's a new model in some ways for putting an oceanographic expedition to sea, we're also trying to maintain consistency with all of our databases.
I will just also repeat and echo the importance of the words consistency and continuous observations, because there have been some studies around the wreck which would provide, a snapshot of biological and environmental conditions. But what we're trying to achieve here is really to build something consistent, and that would allow us to understand a lot of different processes from archaeological perspective, from the degradation of the wreck, and from biological perspective as well. And this is quite unique and only possible because of the contributions of all the expedition partners.
That's great. No, thank you. So a question here, an idea of how long each dive will be and how many we hope to conduct. So each dive runs about 10 hours. It's 2.5 hours to get down to the site. You spend 3 to 5 hours on the site and about 2.5 to get back up. We expect to do somewhere between 10 and 25 dives, and that's really a wide range because of weather. And on each dive, we have a pilot, a researcher, probably one member of this panel or of the science team, and then 3 mission specialists who pay a training and support fee. And everybody has a task to fulfill, whether it's mapping or photographing a certain coral. But then there's an opportunity for just general exploration they can cruise around the debris field and look for other undiscovered objects and the like. I have a question for Steve, which is the role of GIS. How will the GIS mapping help with the future expeditions and the continuing studies?
Well, GIS mapping, at least from my perspective, is a relatively new tool that we've been taking to sea with us for the last, 10 years perhaps. Maybe other expeditions have been using it as well, but so this is a tool that, in one place allows you to assemble a massive amount of data. So all of us are familiar with just standard maps of terrain, but having depth data incorporated into a GIS and into a computer system allows us to manipulate these data. For instance, we can draw 3D maps while we're at sea. These 3D maps will allow us to look for features that normally would be difficult to find. There's a small pinnacle over there that we'd like to explore. There's a depression in the bottom that we didn't see before. All of these things are related to our biological observations. And so it's an incredibly powerful tool. And so on top of these normal map data like depth and bottom type, we'll be able to overlay features of the wreck, mapping of the debris field, the tracks of the submarine, for one thing. And along those submarine tracks, we then have biological data that comes from the video. So because all of these data have timestamps, we can orient them on the map. So when you assemble all of this in one place and put it on a computer screen, it's a very visually captivating and very visually, data-rich tool.
Great, thank you, Steve. A question here about what role does OceanGate Foundation play in the research, and how is it different from OceanGate Expeditions? So OceanGate Foundation is a US-based 501c3 nonprofit that supports the research on the Titanic expedition. So the researchers' travel expenses, equipment that we might need for that, those things are generally paid for by the by OceanGate Foundation through donations to it. OceanGate Expeditions is actually a Bahamian subsidiary of OceanGate Inc., and it handles the expedition component of it, so the chartering of the ship and the crew and things like that. Which brings me into the next question, which is about Horizon Maritime, and a long question, but basically about Horizon Maritime and tug support and other local Newfoundland businesses. We're really proud to work with Horizon Maritime. They've got not only a great bunch of ships, but excellent crews. This expedition is only possible with having the best in all categories, in particular in the ship as well as in all the support. And we had a great success with Horizon Maritime. They showed amazing flexibility in what we do, which is very different than servicing an oil and gas platform or towing an iceberg even, which is what this Horizon Arctic does sometimes in its spare time. So they've been a great partner. We've signed a 5-year agreement with Horizon Maritime going forward to help support this and other missions that are appropriate for its vessels. And then we also have some great partners, and obviously eDNAtec, Subsea Imaging, which is a Newfoundland-based business. We have a fair amount of Canadian content. We have a lot of some of our carbon fiber pieces are made out of Toronto. So there's been a great deal of local support, and we're really excited when we come out this year that our mission specialists and our crew will be able to enjoy St. John's. Last year, we were obviously with COVID, everyone was isolated, so they didn't get an opportunity to wander around town and enjoy everything. So we're really looking forward to that. I also should point out the Miao Phuket First Nations have been a great supporter. We have 2 artists that will be coming along with us to capture sort of the experience side of this. And then we've just completed a Make-A-Wish partnership contest, and we will be announcing the winner of that. So one of the Make-A-Wish supporters will be joining us as well. So it's really been a very local operation, and something that we like to do whenever we leave the United States. And it's been great to have so many qualified and top tier businesses functioning in St. John's. Well, I believe that is the last question. I want to thank all of our panelists for joining us, and everyone who tuned in. This has been recorded and will be available as soon as we've rendered that. And we look forward to coming back again with lots of new discoveries and data from this last